Okay, a lot going on in this slide. What we're going to introduce here in this slide are the myofilaments. So there are 4 proteins that help with muscle contraction. 2 of them are what we call contractile proteins, which are actually doing the contracting. 2 of them are called regulatory proteins, which are actually doing the regulating of the contracting, making sure we don't contract too much or too little. Now, out of those 4 proteins, only one of them is what we call a thick protein, because it's just a lot bigger and thicker than the other 3 proteins, and therefore all the other ones are called thin. But what I want to do first is introduce the 2 contractile proteins. So first, the thick contractile protein is called myosin. And once again, when I'm saying contractile protein, this is going to help our muscle contract. We talked about the 3 things that muscles are really good at. It's excitability, aka irritability, contractility, contracting, and extensibility. This is where that is starting to come into play. Myosin helps the muscle contract and also extend back to its normal place. The other contractile protein is called actin. So actin, you can see, is considered a thin protein. It's a lot smaller. If you take a look at this picture in the lower right corner, you can see actin are these little kind of green circles. And then you can see the myosin are these thicker pink looking things that look like kind of Twizzlers. And then they got these 2 golf club head looking structures. So myosin's a little bigger, a little thicker. Also, it stains thicker and darker on the microscope or in the microscope. So speaking of that, let's introduce how these look underneath the microscope. So because myosin is very, very thick, it is a dark protein. And because actin is very, very thin, it's a lighter protein under the microscope. Now travel back with me to chapter 4 and your histology lab quiz, where you were studying all the different types of tissue, and you had to know the difference between skeletal muscle and smooth muscle and cardiac muscle. And the big difference was skeletal muscle was really, really organized, really put together well, had the multiple nuclei, and it had those stripes. So remember what those stripes were called? They were called striations. And then that's it. We didn't talk about it. We said, all right, those are striations. Let's move on. Now this is where it comes into play, hopefully making some light bulbs go off here. Striations, all they are just the alterations of, or altering patterns of these 2 proteins. So if you take a look at the picture in the upper right-hand corner, what you'll see is this myosin actin just a little bit smaller. These purple structures right here, that is the myosin protein, and then the green structures, the green lines, are the actin proteins. And what you get in your muscle cells is you get this band, this collection where it's just myosin, and then you get this area where it's just actin, and another area where it's just actin. And that continues throughout the entire cell. So we're going to have another area where it's just all myosin in that purple. And then next to that, we're going to have another area where it's all actin proteins in green. So what you're getting is the striations, the stripes. You get a light protein and then a dark protein, and then a light protein and then a dark, and then light and dark and light, and it keeps going and going and going and going, and that's where you see those thousands and thousands of stripes or striations in the microscope. It's just myosin actin, myosin actin, myosin actin. So now we can be a little bit more specific. We don't have to say striations or stripes. We can actually say, oh, those are the 2 altering proteins that just overlap each other, myosin actin, myosin actin. Myosin being the darker one and the thicker one, actin being the lighter one and the skinnier or thinner one. Okay, moving right along. With this picture in the upper right hand corner, I want to introduce the functional unit of muscle cells and therefore myofibrils. It's called a sarcomere. Remember, sarco means flesh, and a sarcomere, as I just mentioned, is the functional unit of muscle cells. And what that means is this is how your muscles actually contract. It's this unit. For example, the functional unit of your brain are the neurons. If you take all the neurons out of your brain, you're brain dead. Your brain doesn't work. The functional unit of your lungs are your air sacs. We'll learn that next semester, but if you don't have air sacs in your lungs, you can't breathe in air, right? And it's to this extreme. So yeah, you die without air sacs, you die without neurons. Your muscles die, or at least they don't work if you don't have these sarcomeres working. So when the sarcomeres contract, the myofibril contracts. And when the myofibril contracts, the muscle cell or muscle fiber contracts. And when you have a whole bunch of muscle cells contracting, the muscle contracts. So the sarcomere, all it is just where this actin and myosin are kind of interchanging and overlapping. And we have these two important landmarks called Z-lines, or Z-disc is a more common term nowadays. And you can see where the Z-discs are, it's just where this actin and other proteins kind of just make this weird zigzaggedy line where there is no myosin. So as I mentioned, one Z-disc or Z-line is the start line of the sarcomere, and then this Z-line or Z-disc would be the finish line. I'm just going to put fin for finish. So what's going to happen is myosin and actin are actually going to bind to one another and pull these Z-lines closer together towards that midline. And when these Z-lines or Z-discs get pulled closer together, the sarcomere gets smaller, or the sarcomere contracts. And once again, when the sarcomere contracts, everything else in the muscle is contracting. So you can picture, if I draw our three myosin bands here and here, and then let's draw some actin. Got our weird Z-disc here, ziggity, 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 and then actin. And then we have some myosin. So here is our another Z-disc in gold. When one Z-disc contracts, for example, when this one contracts with a star, this one kind of also has to contract because it's connected to this Z-disc. So one Z-disc, that's a starting line for a sarcomere. It's also a finish line for the adjacent sarcomere. So once these things start pulling together, they kind of get yanked towards the middle and they all start crunching up and squeezing. And that's how we get this muscle contraction started in relation to the myofilaments, these proteins. There's going to be a lot of steps that lead up to it in just a bit. All right, so almost done with this slide, and then we'll move on. How does myosin and actin bind together? How are these things working together to actually contract the sarcomere? Well, take a look at the anatomy. If you look at the bottom right pictures again, you can see that the myosin has these two golf club looking head looking things, or balloon looking things. And these golf club heads fit really snugly in these holes inside of the actin. So what happens is myosin binds to those holes with actin, and then with the help of ATP actually moves it towards that midline, so the sarcomere, the start line and the finish line actually get closer together. But as long as we had ATP within the area, myosin and actin are kind of like boyfriend girlfriend that just started dating. They're inseparable. They're attached at the hip. All they want to do is be with each other. Nobody else cares or matters in the world. It's just them two. So we need to control that, right? We need to make sure that they're eating and that they're doing their homework and doing their job and hanging out with other parts of their social world, their family members. kind of a bad example, but you get what I'm getting at. So we need to regulate this. Because if you think about it, if myosin and actin are inseparable, if they're always attached to the hip, that means we're always contracting our muscles, which is not good, right? You don't want to contract your muscles 24/7. You're going to have cramp spasms, and you're not going to be able to straighten your joints, and we're going to have all sorts of pain and issues. So we need to regulate this. And this is where these two regulatory proteins come into play. And they are thin proteins, and their names are tropomyosin and troponin. Now, don't get these confused. You might have seen these words before. Tropomyosin is not myosin. It is different. It's a completely different protein. One's thick, one's thin. Myosin is a contractile protein, contracting. Tropomyosin regulates this. Totally different. Some of you may have heard of troponin, or some people pronounce it troponin, troponin, many different ways of saying it. In relation to heart tests, if somebody has a heart attack or some type of heart issue, they'll look at your troponin levels. Totally different. Same pronunciation, same spelling, completely different meaning here, completely different protein that we're talking about here. This has nothing to do with the heart in relation to this slide and to how your skeletal muscles contract. So let's talk about how these things actually work. How do they regulate myosin actin from hanging out all the time 24/7? So tropomyosin is this rope-looking structure that you see blocking, whoops, let me draw that arrow a little better, blocking the actual actin circles. It's that orange rope or that orange string. So if there's a big string in the way, myosin can't get inside of that circle. So they can't get together, and if they can't get together, they can't bind, they can't work. So our muscles are going to relax. Cool. That's great. Except for when we want to contract, then we have to move that rope out of the way, right? That's going to be important. So what in the world is troponin? Troponin is the protein that kind of controls the rope. That's kind of the brains of the situation. So you see this little kind of yellow gold nugget inside of the rope that I'm circling? That's troponin. And the way I always picture this in my mind, describe this to other students, is troponin is the gate or a lock. Well, yeah, troponin is the, excuse me, let's back up. Troponin is the lock or the keypad. Say that we're trying to break into a facility, or we're trying to get into a gated community or something, right? You need to unlock the keypad, and then the gate will open. All right, so the gate is tropomyosin. That's the rope that's in the way. All it does is move back and forth, open, close, open, close. But in order to get inside of that gate, you have to unlock the gate, or you have to unlock the keypad. And troponin is that keypad. So we have to distract the keypad, or unlock the lock, or distract the guard to open up the gate. Okay, last thing on this slide is, what is going to distract the guard, or what is going to actually be the key to our keyhole, or our key to our keypad? How are we going to get the gate open? Well, we have a very, very important element, a very important chemical that serves as our key to our keyhole, and it's stored in the sarcoplasmic reticulum. So if you go back a few slides in your notes, what did we say? What very important ion, very important electrolyte is found or stored inside the sarcoplasmic reticulum? And your answer should be calcium. Exactly. So calcium is what we're going to use to distract the guard or open up the keyhole, whatever analogy you want to use. Then we're going to move that tropomyosin rope or gate out of the way. And then myosin and actin can get together and make the sarcomeres contract, or those Z lines come closer together. So once again, emphasizing the importance of calcium. Calcium is going to help regulate your muscle contraction. So we contract our muscles when we want to, not when we don't want to. So let's move on and keep diving into more detail. You might want to pause and take a little breather after this slide. A lot of information. But once you're ready for to keep going, let's move on and keep discussing this in more detail. So now that we've covered all that material, this slide just serves as a nice little review, putting it all together. You can see that in the top bullet point, actin and myosin are always attracted to one another. This is the new relationship, the boyfriend-girlfriend always wanting to be next to each other. As long as there's ATP present, they're always going to be trying to bind together. And what we call that bind, or that bond that's formed, is a cross bridge. So they kind of just link together, hold hands, and that's called the cross bridge. And I'm going to introduce that in more detail in just a little bit with some drawings, but that's myosin actin. Then we have our gate, or the actual thing inhibiting the myosin and actin from getting to one another, that rope-looking protein called the tropomyosin protein. And then the brains of the operation controlling the tropomyosin, or at least the one that's going to move tropomyosin out of the way, is the keypad, or the keyhole, or the security guard guarding the gate, whatever analogy you want to use. And that's what we call troponin. So these two, tropomyosin and troponin, the two ones that start with T, these are once again called regulatory proteins. They help regulate how much we contract. And then myosin actin, myosin being the thicker one, actin being the thinner one, these are the contractile proteins.